Howdy, my name is Elijah Zavala, and today I'll be presenting on how Nosema apis infections increase winter mortality in Apis mellifera, otherwise known as European honeybees. This project explores how the microscopic parasite contributes to colony losses, particularly in harsh climates. Apis mellifera, aka the European honeybee, plays a crucial role in agriculture by pollinating crops around the world. Healthy bee populations are essential for food production and biodiversity. However, colony losses, especially during winter, have become a growing concern. One of the major contributors to this problem is a parasite called Nosema apis. It's a microscopidian that infects the midgut of adult bees, disrupting digestion and weakening the colony. Infected bees often suffer from symptoms like dysentery and reduced foraging ability. These effects are particularly damaging when bees face environmental stress, such as cold winters. Understanding how this parasite affects colony survival is essential for improving bee health and sustainable agriculture. This leads us to our research question. How does the presence of Nosema apis influence the survival rate of honeybee colonies in harsh climates? To explore this, I reviewed peer-reviewed studies that directly investigated Nosema apis infections, colony health, and seasonal mortality rates in bees. In the first study that I looked at, researchers tracked 50 colonies through the winter, measuring spore counts and survival outcomes. The figure to the left shows their initial data on how brood area and adult bee populations were lowering during the winter cold months, and they wanted to find out why. The results showed a clear trend. Colonies with higher spore loads were significantly more likely to die over the winter. This suggests that Nosema apis infections are a major factor in winter losses, weakening bees when they need to be at their strongest. The second study assessed the impact of wintering methods on Nosema apis infection levels and colony survival, in which they compared indoor and outdoor wintering techniques across the Canadian prairie regions. The key findings of this were that the colonies wintered indoors exhibited lower Nosema apis infections compared to those wintered outdoors, suggesting that controlled environments mitigate infection severity and enhance resilience in the colony. To the right, the figure shows that the blue line, outdoor, has an increased mortality rate as the average number of spores goes up. As compared to indoor, it's much lower. The third study examined how Nosema apis infection impacts the physiological health of bees. Researchers analyzed the bioelemental content such as sodium and iron in Apis mellifera during the summer and winter months, comparing infected and uninfected individual bees. The key findings of this study was that Nosema apis infections led to significant deficiencies in essential bioelements, particularly during winter. These deficiencies are linked to increased mortality and accelerated aging in Apis mellifera, highlighting physiological stress imposed by Nosema apis. On the figure to the left, you can see that the top left and top right are dedicated to Nosema apis free bees, and on the bottom are Nosema apis infected bees. And then on the left, it's representative of winter, and on the right, it's representative of summer. So from the figure, we can see in the bottom left, which is resemblant of infected bees during the winter months, you can see that they are at their lowest bioelement availability, and therefore it's a compound effect with these winter months and Nosema apis. In conclusion, we can verify three main points. Nosema apis infections are positively correlated with increased winter mortality in Apis mellifera colonies. Secondly, indoor wintering methods reduce Nosema apis infection levels and enhance colony survival compared to outdoor wintering. Third, infected bees exhibit significant deficiencies in essential bioelements during winter, contributing to higher mortality rates. With those 3 points in mind, we can see that Nosema apis is a contributing factor to high mortality rates in the winter and definitely add a compounding effect. These findings have real-world implications for beekeepers. Knowing that Nosema apis infections can drastically increase winter losses, especially in harsh climates, gives us direction for prevention. Indoor wintering Regular parasite monitoring and nutritional support may help reduce mortality in these bee colonies. And for future research, more controlled longitudinal studies are needed to explore how environmental stressors like cold temperatures interact with Nosema apis infections. Secondly, the development of early treatment tools such as probiotic supplements or antifungal agents could help mitigate infection before winter stress even sets in for these bees. Here are the peer-reviewed sources that informed my presentation. Thank you for watching. I hope this gave you insight into how one microscopic parasite can have such a significant impact on honeybee survival in cold climates, and eventually, our agriculture and our food.